a pleasure to be here at AGBT this year to present at the Illumina Gold Workshop. And I'd like to thank Illumina for this great opportunity. For the last dozen years or so, my lab has been really focused on developing cell-free DNA methylation liquid biopsies to answer important questions in cancer. And we spent a lot of time on ovarian cancer risk assessment. And over the past year or so, we've worked closely with Illumina to evaluate how their newer multiomic platforms could help address some specific methodological gaps that we've encountered in plasma resolution. These are my disclosures. The clinical challenge for women who present with adnexal mass, which is a mass that arises from the ovary or nearby structure, is determining if a mass is malignant or benign before surgery. Biopsies are not safe for this patient population. And so for this reason, women face a high-stakes triage decision whether they should undergo surgery with a gynecologic oncologist or be managed more conservatively. Current risk assessment tools are protein-based. They achieve reasonable sensitivity, but specificity for benign disease remains quite limited, leading to high false positive rates and unnecessary referrals and potentially avoidable invasive procedures. So the central question that we asked was, can we develop a plasma-based test that maintains strong cancer detection while substantially improving specificity for benign disease? To address that question, we developed a test called OvaPrint, which is a cell-free DNA methylation-based risk assessment classifier using hybrid probe capture and bisulfite sequencing. In our first study, OvaPrint achieved 84% sensitivity and 96% specificity. The development work initially focused on high-grade serous ovarian cancer, which is the most common and most lethal subtype of ovarian cancer, and then subsequent studies expanded evaluation to additional histologies. In a prospective validation cohort, OvaPrint continued to achieve excellent specificity at 96% against benign disease, and the high specificity translated to a 2 to 4-fold higher positive predictive value than CA125. Sensitivity in this study was 100% for high-grade serous ovarian cancer, which is the most lethal subtype, and supports accurate identification for this disease in this cohort. However, sensitivity was lower for the non-high-grade serous histologies in plasma, and this raised the question of whether the reduced detection reflected a signal resolution limited in plasma. When we applied reduced representation bisulfite sequencing to tissue genomic DNA, you can see in the upper two plots that the benign and malignant samples separated extremely well across all histologies. However, in plasma, cell-free DNA separation was reduced for non-high-grade serous ovarian cancers. And this indicated that the underlying biology and epigenetic signal was present in tissue, but attenuated in plasma. We therefore asked whether improving methylation chemistry and incorporating orthogonal biological layers could enhance signal recovery and sensitivity for these samples. To address the signal resolution question in plasma, we expanded the analysis across orthogonal platforms. We used the Illumina 5-based chemistry, which we evaluated before and benchmarked, and demonstrated improved genome complexity and greater usable methylation data. In parallel, we profiled over 9,500 plasma protein using the new Illumina protein prep to capture circulating protein biomarkers associated with tumor biology. We also incorporated Illumina's Spatial Transcriptomic platform to examine tissue architecture and cellular context. So together, these approaches interrogated circulating methylation and protein biomarkers alongside tissue organization, allowing us to assess the alignment across biological layers rather than relying on a single biomarker class. Using the 5-based chemistry, we observed improved separation of the non-high-grade serous ovarian cancers from benign samples and cell-free DNA. And importantly, you can see on the right side that the OvaPrint false negative cases began to shift away from the benign cluster for the first time. And we were very excited about this. So the better chemistry, we believe, does preserve more informative CpG states and reduces the stochastic loss of signal in these low input samples. And this could directly improve the sensitivity without compromising the specificity. On this slide, you can also see separation between benign and malignant adnexal masses at the protein expression level with the Illumina protein prep, including among the cases of the false negative of OvaPrint. These findings demonstrate that the plasma protein profiles can provide additional biological information in this setting and can be used to further develop biological and biomarker strategies for women with adnexal masses. But the other thing that it can do is it can help us understand the biology. And here you're looking at individual proteins that show upregulation in invasive cancers compared to borderline tumors, which are non-invasive cancers and benign adnexal masses. And the multiple proteins demonstrate, you can see from the graphs, a distinct shift in the expression in these malignant cases compared to benign. Here we take the differentially expressed proteins and evaluate them at the pathway level, and we did this analysis in ICM, and in fact we did a lot of these analyses in ICM, and it was wonderful to be in one place to do it all. And the enrichment analysis here shows that these proteins group into defined biological programs associated with ovarian cancer that are known, but also new things that we didn't know before that we will be investigating further. So instead of looking at just the individual proteins in isolation, we're seeing coordinated biological processes represented in the plasma. Next, we interrogated the 5-base methylation data with the plasma proteomic data using ICM. In one example, in the black box, an OvaPrint false positive case was resolved when the methylation and the protein signals were examined together. So across the cohort, the combined analysis didn't materially change overall malignant benign separation, but what it did allow us was seamless cross-modal interrogation within a single framework. We were able to evaluate methylation and proteomic data together quickly and without moving between disconnected systems, and this made the interpretation more efficient. Using Illumina's new spatial transcriptomic platform, we examined the tissue architecture and gene expression patterns of a benign cystadenophybroma, which was also negative by OvaPrint. The spatial data were consistent with benign histology and showed no expression of cancer-associated markers identified in the plasma analysis, which are the purple spatial plots that you see. And this provided tissue-level context that supports the plasma classification, and by the way, it's also the first report of a spatial transcriptomic analysis of a benign adnexal mass. In this one, we applied spatial transcriptomics to a high-grade serous ovarian cancer sample, and despite lower quality tissue, the spatial patterns aligned with known high-grade serous ovarian cancer biology and also supported its fallopian tube origin. The regions showing malignant features corresponded with expression of the proteins that we identified in the plasma data set, and that's what the green dots that you're seeing are. So this demonstrated concordance between tissue architecture and the circulating signal. In this spatial transcriptomic analysis of a benign mucinous cystadenoma, this was classified as a false positive by OvaPrint. The spatial analysis did confirm its mucinous histology, but revealed immune infiltration and low-level expression of certain cancer-associated markers, likely explaining why it was a false positive. This case was the one that was resolved alongside the 5-base methylation and proteomic data set. So the spatial layer here provided architectural context that helped interpret also the plasma findings. So OvaPrint has high specificity for benign disease and sensitivity for high-grade serous ovarian cancer. The addition of the 5-base methylation improved signal resolution in plasma, particularly in the non-high-grade serous cases. The proteomics analysis provided complementary biological information at the systemic level. Spatial transcriptomics added architectural context at the tissue level. So while integration did not materially change overall cohort level separation, it strengthened case level interpretation and provided biological coherence across the modalities. Importantly, in the remaining 3 false positive cases that are pointed out, the data suggests that underlying biological complexity rather than a simple misclassification. We'll be examining those further. So in summary, I'd just like to add and just finish by saying that conventional cell-free DNA methylation methods may lack sufficient resolution in plasma, and that the Illumina 5-base improves that cell-free DNA quality. The 5-base derived methylation signals did enhance in our study separation of a very stubborn set of tumors that were traditionally hard to separate, and plasma proteomics and spatial and transcriptomics sort of tie the story together and provide this comprehensive picture that you can do in a seamless framework now available.